Hey guys, welcome back to another lecture video for Chem 104. In this lecture video, we are going to talk about the Fischer projection for monosaccharides. And so Fischer projections were invented by the scientist, his name was Emil Fischer. And so Emil Fischer worked with, the stereochemistry of a lot of carbohydrates, and eventually he kind of came up with a model, a very simple model for drawing compounds with stereoisomers in a very efficient and precise way. And he did it specifically for carbohydrates. And because of his accuracy for being able to predict the stereoisomer for carbohydrates, through his drawing, he was able to get a Nobel Prize for it in 1902. And so basically what he is doing in his model is that he's taking a three-dimensional representation of, for example, a carbohydrate in its linear form, and he's representing it in a two-dimensional manner. However, he is doing it in such a way that it represents the it's more or less like a shorthand notation to represent the three-dimensional geometry of those carbons that have stereoisomers or chiral carbons. And so before I talk about these simple rules, I just want to kind of review the tetrahedral geometry of carbons and its depiction using different types of lines, different types of covalent bonds. And so since a carbon is tetrahedral in terms of its electron geometry and molecular geometry, there's actually several ways that you guys can depict these tetrahedral carbons. And so one of the most common ways is to have two bonds that are in plane with the central carbon, and that's why these are like normal lines, the lines that we usually use to represent a covalent bond. And so if you have two normal lines, then that means that you have one bond that is going to be above the plane of the central carbon, and you have another bond that is going to be found below the plane of the central carbon. And so this perspective is us looking at the carbon like face in front. However, there's another way for us to visualize this carbon such that our eye is focused in between the bold and dashed lines. And so if we kind of imagine our perspective through this path, we get a central carbon in which we get something that's bold, meaning it's above the plane of the carbon, and this dashed line is going to be represented as another bold line. So remember, the perspective is changing. Our eyesight is peering through, our line of sight is peering through the middle area of the dashed and the bold. And so if this is our line of sight, then that means this is still going to be bold, but now this becomes bold as well. The dashed line becomes bold as well. And so if this is our line of sight, then that means that these two normal lines are now going to become dashed. Okay? Because they're, with respect to our perspective, our line of sight, these two bonds are now behind the plane of the central carbon, while these two bonds are pointing towards the eye. Okay, so that's why I'm representing them as two bold lines, and these two normal lines become these dashed lines. Hopefully that makes sense. Anyways, so the idea is that everything that is a dashed line for the Fischer projection is going to represent a vertical line. So vertical lines under the Fischer projection is representing the bond that's going behind the plane of the carbon. And so the bold lines, or the bold covalent bonds, in the Fischer projection is going to be depicted as a horizontal line. And where the vertical line and the horizontal line meets, that's going to represent a chiral carbon. And so this is basically the premise of a Fischer projection. Our perspective is that we're seeing two bold and two dashed. And we're going to represent those bold and dashed lines as either a vertical or a horizontal line. Okay. And so vertical lines, once again, are representing the dashed lines. And dashed lines means that those bonds are going behind the plane of the carbon. Horizontal lines are wedges. Wedges are bold lines, if you will, and they're going to be above the plane of the carbon, right? So it's pointing at us. And so an example of this is glyceraldehyde. So I'm just going to kind of zoom in here. And so if we take this very, very simple carbohydrate, this 3-carbon carbohydrate, by the way, just as a way of review, this is aldo trios. There's 3 carbons. So these are 1, 2, 3 carbons. There's an aldehyde. So this is aldo trios if you want to classify your sugar. Anyways, so basically we are going to represent, excuse me, these carbohydrates very specifically in a Fischer projection. So first, we want to make sure that our aldehyde or ketone is going to be as high up as possible. Okay. And anything that is dashed is going to be represented as a vertical line. And anything that is bold is going to be represented by a horizontal line. So Emil Fischer is taking these he's taking these 3-dimensional structures and he's creating a 2-dimensional structure out of it. And so the Fischer projection is literally what you guys see here. Okay. And so where the bold and dashed lines intersect, that's going to be where the chiral carbon is going to be located. And so it's very quick, it's very efficient. Anybody can draw vertical and horizontal lines very quickly. You don't have to, write every single letter down to represent the carbon, to represent the oxygen and the hydrogen. So it saves time. And so the strength of the Fischer projection, besides the efficiency for us to depict carbohydrates, is the fact that his representation can predict the overall stereoisomer of the carbohydrate. And so remember, this is in 1902, where there's not, there's some advances in technology, physics definitely, but they don't have these machines that we can just quickly plug things into, and the computer will tell us whether or not it's rotated, whether or not the polarized light is going to be rotated clockwise or counterclockwise or whatever, right? So 1902, this is before the dawn of computers, if you will. And so, his structures was able to accurately predict the stereoisomer of carbohydrates by looking at the last chiral carbon that's farthest from your functional group, either your aldehyde or your ketone. And so, long story short, if the last chiral carbon that's farthest from your aldehyde or ketone, if your hydroxyl group, and I'm going to change colors here. So if your hydroxyl group is found on the left-hand side, then the L stereoisomer will be observed. If the hydroxyl group is going to be found on the right-hand side, then your D stereoisomer will be observed. And so his Emil Fischer's projections was able to accurately predict this using very, very simple rules. And that's why he got a Nobel Prize for it in 1902, just by drawing lines. Anyways, so let's go ahead and look at how this is more prominent in larger carbohydrates that have multiple stereoisomers or multiple chiral carbons. And so looking at glucose, everything that has an asterisk is a chiral carbon. And so first of all, let's understand why they're chiral carbons by looking at some of these carbons that's been marked. And so if we're looking at this carbon, we need to understand why this carbon is chiral. Well, if you look at the top, it's a CHO, so that's an aldehyde. That aldehyde is different than an alcohol or hydroxyl group. And that aldehyde and hydroxyl group is really different from this hydrogen. And all three of the things that I circled around the central carbon is different than everything that I'm circling at the bottom. And so therefore, this carbon is going to be chiral. Oops. Okay. So let's go ahead and take a look at the next carbon. And so looking above that central carbon, notice how that piece that I circled is different than this piece that I'm circling, which is different than this piece that I'm circling, and this piece that I'm circling. So if I have 4 different substituents or 4 different items attached to that carbon, then that carbon is going to be chiral. It's going to be a chiral center. And so you guys can, play with that logic on your own. But long story short, what this Fischer projection does is that it assumes that everything that's vertical will have dashed lines, if you will. And everything that is horizontal, I'll do that in blue, is going to have that bold line or that wedged line. So this is telling you that these hydroxyl groups and these hydrogens that's found on the left and right of this glucose molecule are pointing up. They're pointing towards you. And everything else is going behind. And so, once again, everything that is vertical represents this dashed line, which is what we see here. And everything that is horizontal is representing the bold lines. All right, anyways, back to my main point. The Fischer projection is able to accurately predict the overall stereoisomerism for these larger carbohydrates with many chiral centers. So if you have many chiral centers, such as glucose, what is the overall stereoisomer? And so that was a big thing back in the day. And so basically what Fischer said was that the last carbon that's chiral, that's furthest from the aldehyde or the ketone, if that hydroxyl group is on the right, then this is going to be the D stereoisomer. Okay, and so if you have a carbohydrate such that, so remember these are all chiral centers. And so let's just say I have OH, OH, OH. And so he basically said that the last chiral carbon, which I'm going to go ahead and mark in pink, that's furthest from the aldehyde or the ketone. If the hydroxyl group is on the left-hand side, then this is going to be the L stereoisomer. And it turns out it's true, right? There were experimental studies that later confirmed this, and these studies was able to validate his hypothesis and validate his projection, his drawing, his model. And because of the simplicity of his work and what it can prove, he got a Nobel Prize for it in 1902. And so I'm going to go ahead and show this specific slide. And so these are all ketones, by the way. And so I'm going to go ahead and mark this in pink. So notice that your carbonyl carbon is wedged in the middle of a carbon chain, not found at the very end of a carbon chain. So therefore, this is a ketone. So all of these ketones or ketoses, they're going to act as our point of reference. And so the last chiral carbon which I'm circling in green, that's farthest away from the ketone, that's going to dictate whether or not the stereoisomer, the overall stereoisomer for this carbohydrate is going to be D or L. And so once again, since these hydroxyl groups are on the right-hand side, all of these molecules are going to have this D stereoisomerism. And so it turns out that these stereoisomerism is important once again in our bodies. And so if you guys recall from the previous lecture video, our proteins that's inside our body are very very specific to the types of substrates that is allowed to come in. And so they're stereospecific, meaning that only substrates with very specific stereoisomerism can interact with very specific proteins, if you want to just generalize it in that manner. And so the enzymes in our body that utilizes glucose, that breaks down glucose and extracts electrons from it to later on create energy in the form of ATP, they can only recognize the D form of glucose, the D stereoisomer of glucose. And so the L stereoisomer of glucose is not recognized by our enzymes. And so that's pretty interesting, right? So if you guys ingest synthetic L-glucose, oops. So if you guys ingest synthetic L-glucose, our body doesn't have the enzyme to recognize this specific stereoisomer of this carbohydrate, and therefore it cannot utilize it as a source of energy. It cannot break it down, it cannot extract its electrons. There is, however, one exception, and so this is pretty interesting. So this type of plant pathogen bacteria, I'm not even going to pronounce its name, has an enzyme that can oxidize L-glucose. And so this is our first example of an organism that can utilize the L stereoisomer of glucose and be able to break it down and use it as a form of energy. And so if you guys ever study bacteria in greater detail, you guys will probably walk out of that class or out of that semester having, like greater appreciation for the diversity of bacteria. They are the most adaptable, organism. I personally feel that they're the most adaptable organism. And so if you look at the evolution of man, we're pretty much stuck in what our enzymes are capable of recognizing. But with bacteria, here we can't even utilize L-glucose, but this bacteria can. And so these bacterial organisms are pretty interesting. Okay, and so the next lecture video is going to be Haworth structures, and we're going to learn how to go from a Fischer projection, and we're going to learn how to cyclize it and produce what's known as a Haworth structure. See you guys in the next lecture video.